Our moderator is Dr. Donald Kearns. Many of you will know him. He is the chair of the board of trustees for Sanford Burnham Prebys and was formerly the CEO and president of Rady's Children's.
Wow. Are we all going to the gym after this? I hope you all are as excited as I am to be here. It's incredible. I've had the opportunity to spend a lot of time with this group of geniuses up here. These are the people that you used to copy off of during the math exam. And just listening to what they do is incredibly exciting. And I just want to let you all know that the only reason I'm up here is they wanted to average out the IQ down here. So why don't we get started? Paul, from a research standpoint, where are you seeing the most promising convergence of basic discovery and patient care?
Yeah, that's such a great question. I think all of our talks reflect where we're seeing a huge impact of research in clinical practice, which is the ways in which people live their lives. We talked about exercise, we talked about diet and sauna. We didn't even talk about sleep, probably the single biggest determinant of health and well-being. And we're identifying a huge range of ways in which modifications to lifestyle can impact disease and how bad it is and how it can be cured, but also prevention and ensuring that we have long, healthy lives. At the same time, there's so much data. We all talked about robotics and DNA sequencing, and that means the tools of statistics, data science, machine learning, AI have become foundational to biomedicine. Biomedicine is now a data science, a discipline made of data, and all of us spend just as much time thinking about the complicated data and how we translate that into clinical practice in an easy, seamless way.
Thank you. And if you can help us, what's the role of inflammation in Alzheimer's? And perhaps we can also talk about cancer too and inflammation.
Right. So I like to think of inflammation as a double-edged sword. Your immune cells in your brain are really important for fighting off infections and clearing away debris. But if they're overactive, that's very bad too. And so when sequencing of the human genome started to happen and a lot of genomes were sequenced, it began to be understood that there were genetic variants in immune genes that led to Alzheimer's disease. So it's clearly a really important component of Alzheimer's disease. And I think also important as a therapeutic target, like some of the drugs we talked about today.
And in cancer, it's a related but a little different story. In a deep way, most of the things that you think about as causing cancer actually cause inflammation. Obesity, smoking, alcohol, each of them lead to a chronic, long-term, low-level aggravation of the tissues. And when tissues get aggravated for a long time, they get unhappy. And that response to the unhappiness is often the development of a cancer. The cancer will be almost the body's response to how do I get rid of this stress? It's by getting rid of normal processes that make it recognize the stress, and those normal processes are the ones that give rise to cancers. So in a deep way, early inflammation is one of the most important causes of cancers. And so genes that make people more sensitive to inflammation also tend to make them more sensitive to cancer.
Thank you. Sanjeev, when you look at the future of cardiac research, what breakthrough feels closest to becoming reality? And how would it change the way we treat heart disease now?
So when I think of breakthroughs in biology right now, Paul alluded to this in his talk as well too. But what I think all of us are seeing and hearing, and you just every time you turn on the TV, you listen to the radio, you hear of how much AI is taking over every single aspect of what we're doing, right? And this is true in biological research. It is absolutely amazing how much in the past 3 or 4 years, biologists are talking about AI at the bench in the things that we're actually doing. So when I think about the discoveries and the breakthroughs in the cardiovascular sense, I think about it kind of more broadly from all of biology itself too. You know, the first sort of iterations of how AI was used was to try and understand the atomic structure, what every single protein in our body looks like. And that's now morphed into an ability to basically predict what mutations could be the most important for disease, and just actually then take an entire cell in your computer and model how that mutation might actually be happening. We could advance, like what Anne was talking about with repurposing drugs and trying to accelerate the translation process. This could be a way that we could advance the entire scope of what we're looking at to test the most important things very, very quickly. I think in the context of cardiovascular disease, heart failure and what happens right after you have a heart attack and how you can potentially clear the scar that forms and also get your heart to be healthy again, that right there, the applications of AI to that is a huge, huge field. I study pediatrics, so when we think about what is one of the major obstacles for us, it goes back to that pie chart I showed in the very first slide. We just don't know what causes half of these heart defects in these little kids, right? Why are they coming into the clinic born with a hole in their heart, right? And I think this is where applications of AI could actually revolutionize how we could start to think about what could be the causes of this. We don't know even where to look right now. And this is, I think, one of the biggest breakthroughs. Paul, I know you see this in your field as well too, right?
Yeah, definitely. I think one of the most notable things we see in AI is how much more efficient and faster it makes research. I'll give an example. I have a lot of people on my team who are software engineers or programmers. And when you get a new person out of undergrad, they might write 100 lines of computer code a day. My best people who are coming from the major tech companies, senior, they write 1,000 lines of code a day. Not anymore. Using AI tools, 10,000. 10,000 lines of very high quality code because there's this acceleration adding a zero to the productivity. So it becomes, how do we capture that? How do we help people learn to use these tools effectively while keeping the core human innovation that drives really exciting research?
Caroline, everybody I know seems to be on GLP-1. If we define aging as the gradual decline of metabolic resilience, do GLP-1 therapies meaningfully alter that trajectory?
Yeah, I get that question a lot. As you said, a lot of people are on these, and I'm sure everybody has seen the amazing effects on weight using these GLP-1 therapeutics. And you haven't, just tune into the Oscars in a couple of weeks. But, so what these drugs are really, really good at is management of diabetes, reducing inflammation, and of course, weight loss. And then in population-based studies, what we're also seeing that they're really good at reducing cardiovascular diseases and strokes. So the predisposition to the development of some of these most dangerous age-related diseases is definitely reduced by the use of these GLP-1 therapeutics. So that's really a wonderful outcome of all of these diseases. One caution is we don't have long-term studies yet. They have been developed really recently, and we don't have long-term outcome studies. And one other thing is that we have seen that a lot of muscle loss is occurring when people are on these GLP-1s. So when you're on these, you have to be very careful in counteracting these and keep on exercising. We keep coming back to the exercise thing, but keep on exercising so that you are not prone to all of that muscle loss, which then could lead to more frailty. So are those magic aging cures? No, but they can definitely reduce some of these risk factors for the development of age-related diseases.
You may be getting the gist that Equinox is our new partner. With diseases, and with diseases like Alzheimer's, early detection is so important. Are there any early detection methods like blood tests?
Yeah, so relatively recently, there's been, I think, two early detection methods that are blood-based, and they're detecting the proteins that we talked about today, tau and amyloid beta. that have been approved. So they're not used widely across the public. They're used to confirm Alzheimer's cases for suspected cases. But it's a really exciting development. And one of the most exciting things about it is with diseases like Alzheimer's, the earlier you can intervene, the better, right? If you wait until the point of no return, it's a no-go. So having these sort of tests where we can identify people way before they have symptoms is really important. Of course, it has to be paired with the tests have to be accurate and reliable and work across different people and different ages. And we're looking forward to the day when the tests are actually paired with effective therapies.
Yeah, and if I may add, so we are now able to measure the cellular recycling process that I was telling you about in blood samples. And what we have found in Alzheimer's, or what has been found in Alzheimer's disease, is that the cellular recycling process in the brain is deregulated or dysregulated. So what we now want to see whether we can detect that this process is also deregulated in the blood, because if we could test whether it reflects the brain status of this process, maybe we can link and use this also as an additional biomarker. And what is so good about using blood for biomarkers is it is so accessible. Everybody is able to go to the doctor and give a vial of blood. So if we're able to really further the development of these biomarkers, I think that is a really hopeful strategy of early detection.
Yep, much better than a spinal tap.
Paul, we seem to have more and more friends diagnosed with cancer. And years ago, that seemed like a death sentence. And more folks, it seems to be like a chronic illness. Is that something we're going to see more of? Do we see treatments dealing with that? How does cancer become a chronic illness instead of a death sentence?
Yeah, it's a great question. And it actually goes back basically 60 years ago when the US government decided to create the National Cancer Center program and created these centers of excellence. And the first thing that everybody asked was, why does cancer happen? What's going wrong? And there were these decades, 40 years, of finding the specific genes important in cancer. And that was a lot of work, but then once we knew them, we could start deploying drug discovery techniques, now today AI tools, drug repurposing like Anne discussed, to be able to find therapies. And those therapies can be, in some cases, remarkably effective. There's a story I love. This is always my favorite cancer story, because there's a type of cancer called promyelocytic leukemia. It's a blood cancer, PML. When we identified the genetic cause of it, it turned out to be related to vitamin synthesis. So what was the treatment? They tried just giving people a very high dose of a specific vitamin. Ran a clinical trial with 50 people. They had to stop it early because 49 were completely cured. Not every cancer is like that. I wish that vitamins cured every cancer, but they don't. But it gives you an example where understanding the basic science and cause of cancer allows us to design very precise, very targeted treatments, sometimes drugs, sometimes interventions about our lifestyle. And those things can take a disease like breast cancer, which 50 years ago, the median survival was 5 years. Half of women died in 5 years. To today, 25 years. That slow incremental process of basic research is what's doing that for cancer. And it's what's going to eventually take it all the way.
So this question is actually for each of you individually. What's the boldest question your research is trying to answer right now? What gets you up and excited about driving in the morning? Why don't you start, Sanjeev?
Boldest question that my lab is trying to answer is to try and take an approach where, you've heard from almost everyone on stage about how we can sort of form a relationship between clinical research and basic research, right? So how can we actually find insights from the cells of the individuals who have the disease, right? And so one of the things that my lab is doing is you've seen in my work and in the work of what Anne presented as well, the ability to take skin cell, convert it back into a stem cell, and then turn it into all types of cells in our body, right? What we can do now is instead of making just like a two-dimensional layer of cells, we can now try to make a three-dimensional organ in a dish. It's not quite the real organ, it doesn't quite beat, but if you come to my lab, in which I hope every single one of you would, you can see little beating blobs of heart cells in a dish, right? And so we call these organoids. These are three-dimensional representations of the organs in our body, right? They're not perfect, but they're starting to mimic the actual structure and the morphology of the organ, right? And we think that that might actually be a way to more quickly go from insights that we learn at the bench, what genes are important, what chemicals might be doing something useful. But instead of having to go all the way into a mouse or some other animal study, which takes so many years and costs so much money, can we go straight away into something that looks like an actual human organ and ask, is this thing really working? I think that's one of the boldest things that we're doing. And one of the things I want to say immediately is that the way to do this is with collaboration. So I think all of us are talking about how we can work with collaborative options with folks who are in the clinic. And I'm so happy to say that we are beginning collaborations with folks, clinicians at Rady Children's. One specifically is a pediatric cardiologist, Dr. Paul Grossfeld, who collaborates with my lab on a project called hypoplastic left heart syndrome. It's this really severe form of heart defect. And so we've literally taken cells from a person that Paul is treating and now can make little beating hearts in a dish in our lab. So that's one of my boldest things.
Thanks. Caroline.
For me, the real goal is to close the gap between health span and lifespan. In the US, especially women live 13 years in poor health at the end of their lives. 13 years, that's a long time. So what I like about being in the aging research field is that we're going away of thinking about what goes wrong, but thinking about the interventions of how we can make things better. And what I'm excited about is really understanding how these interventions work. So how does exercise, how does sauna, what are the molecular mechanisms that are actually activated by these interventions so that we can then harness those to improve lives and health in especially prevent aging and live healthier lives.
Anne.
Yeah, so what keeps me coming into work every day, I'm a stem cell biologist, and we use stem cell biology to create models of neurological disease to look for disease cures. And the really powerful thing about these models is that we make them from individual people, that they have their own genetics. And I think one reason that there's been failures in clinical trials is that there's been this approach that one size fits all. You make one drug and it's going to work for everyone with the disease. That's not really the case. There are many different types of Alzheimer's, for instance. And so we can learn to understand the genetics of individuals and better match them with the right drug. I think it's really going to be revolutionary in the future. And so the only way really to study human genetics, which are very complex, is with these human cells.
And Paul.
I love that all our answers have come down to making research and medicine personal. How can we take the latest technologies, innovations, and figure out what's the best way to do it for me? I cannot stand being on a treadmill. I hate it. Can I do weights? Can I just play sports? Can I play tennis? I'd rather play tennis. I don't know. That's definitely the most exciting question for me every morning. But I also have another hat at Sanford Burnham Prebys. I'm the vice president for data science and AI. And in that context, the other thing that gets me up every morning is to think, how do we use these tools the right way? How do we make sure everybody in biomedicine is using them to make our research faster, more efficient, more effective, and still ethical, still ensuring that we are making sure that we maximize the value of it without compromising our values? How do we teach the next generation to use these tools, but to use them as an adjunct, not as a crutch, to make sure that it's supporting innovation and not replacing education? And so I always think about that every morning, first thing. How do I use AI properly? And that usually starts with looking at the weather.
Okay, so last question for you all. You spent a fair amount of time talking about how important collaboration is, and Sanjeev got started there. The future of science, I would imagine, especially with the amount of data that is being produced worldwide now, is going to be about collaboration. Walk us down that road. Tell us a little bit about who you're collaborating with now, and what do you picture happening from there? Sanjeev, why don't you start?
So Paul mentioned that he's the vice president. He's in this new data science and artificial intelligence center that is now formed as part of David's vision for how we can bring Sanford Burnham Prebys into the absolute latest era of biomedical science. I'm also part of this center. I collaborate with many of the folks in that center. Dr. Sanju Sinha is a close collaborator of mine, and he is the person who has generated and developed so many new methods for just simply looking at images of healthy and diseased tissue and being able to simply extract information from that, just from an image, right? My student, I mean, the pace of what is happening right now is so incredible. I had a student join my lab who had some ability to do code, to understand computational biology, but very little, and she had no ability. She had never worked on AI projects before. In less than 2 years, we're about to write our first paper on a brand new method that we've now just invented that is looking in collaboration and in inspiration from what Sanju's done of looking at images of healthy cells and disease cells and being able to connect it with all sorts of new kinds of technologies, right? That's something that I would never have thought was possible. And that is possible in a small institute like SBP, like Sanford Burnham Prebys, where I can just call somebody up, walk down the hall, and talk to somebody who knows so much more about this and immediately strike up a collaboration.
We have a collaboration.
Yeah, we're collaborating right next to you.
Yeah, I like what you said also about your student. I think for me, collaboration is also the collaboration with the people in our lab. Mm-hmm. The education and the impact that we have on science is amplified through the people that we train. So I think training the next generation of scientists is very, very important. And we all do that in our labs and through our roles with the graduate school. Other than that, for collaboration for my lab, the assays that we do in our lab are very specialized and not a lot of people are able to do them. So we're getting either approached of doing assays for other people who want to incorporate that into their studies, or we have also trained people from, for instance, we had visitors from USC coming down that were then trained for a couple of weeks in my lab to bring it back. So yes, it is a team science, and we're happy to share our knowledge and improve scientific knowledge all across the world.
Yeah, so my lab, our projects are very collaborative, and the projects that I spoke today were both major collaborations, one with the Epstein Family Foundation. There are sort of two types of collaborations that I have. One is multidisciplinary, and these are large projects that involve people from all sorts of different areas of research. So one I'm on right now, there's clinicians, geneticists, informaticists, people like me that are cell biologists, drug screeners. The other type of collaboration that I've been involved in, part of my lab works on rare disease, and that's collaborations with foundations that support this type of research. And it includes the parents of the children with their cells, the physicians that work with these children. For my research, it's indispensable, and it really enhances the way that we can do our studies.
Oh, I think in cancer, in oncology, the most fundamental collaboration is always with the patients. We have incredible resources of cancer tissues, blood from cancer patients, images from cancer patients that have been donated for research purposes. And without that, 90% of what we talk about in cancer wouldn't be able to move forward. It's truly essential. But even if you take a look at the research that I talked about, it's intrinsically a team sport, a global team sport. Like hockey.
For instance.
I couldn't resist. So, but the patients on the exercise clinical trial, some were from City of Hope, some were from Memorial Sloan Kettering in New York. If I look at the DNA sequencing studies that I talked about, those were people in Toronto, Melbourne, Singapore, UCLA, like all across the world. And it is not possible to do science today without that global collaboration and the engagement of patients to help us push forward the most impactful questions.
David, we're so excited to have you here. I'm serious. David and I have been colleagues for many years, and he's a dear friend and a genius, and has been able to recruit incredible scientists from across the globe.
As you say.
Tell us a little bit about your vision of the future. What do you
I get the last word?
You get the last word.
Wow, thank you. Anyway, so I am so happy to have these people as my colleagues. If you ask me why I get up every day and excited, you just heard it. I mean, it's have amazing research. And people at the Institute, they ask really hard questions, really important clinical questions. You can do research at an easier level and do some things that derivative. But as you heard, these are all groundbreaking, first and done. And so this is really quite different. So my job, I really think, is to create an environment for my colleagues to do really well. So it's to get the resources, it's to find the bottlenecks and fix them. It's what's equipment, what's the state-of-the-art requirements, and then to create an environment of collegiality and collaboration where everyone can work together. It's team science to do these really difficult problems. So with that, I think there are probably 100 more questions. So I am going to recommend that we have a reception. I will not let any of them leave to get the answer to all your questions. And I think they deserve a round of applause. You all for joining us.
Please search these guys out. They won't bite.
That's right.
And they'll answer all your questions.
Thank you.
Thank you.